Good morning, students. Today in this lecture talk, I shall discuss on first B.Sc. second semester microbiology paper, syllabus of both theory and practical, affiliated to BCU, of subject B.Sc. Biotechnology, NEP batch. Okay, now let us go through the syllabus of B.Sc. Biotechnology, basic or honors second semester. Okay, course 1, theory code, B.Sc. T2 BT C102 microbiological methods. Okay, so total 56 hours theory you need to attend. Unit 1, instrumentation, where you are studying microscopy, in that principles of microscopy, resolving power, numerical aperture, working principle and applications of light, compound microscope, dark field microscope, phase contrast microscope, fluorescence microscope, confocal microscope, electron microscopes, TEM and SEM. Okay, all these different microscopes you are studying in your syllabus. Be familiar with those microscopes. Further, you are studying analytical techniques. You need to understand this analytical techniques in detail, working principle and applications of centrifuge, ultracentrifuge, spectrophotometer, chromatography, paper and TLC. Okay. Next, further unit 2, sterilization techniques. I hope you are making note of these units. I request you all to, in your notebook, first page, kindly make a note of all these units. Once we finish syllabus, you can just tick that one as it's completed, like that. Okay. Unit 2, sterilization techniques, where in the beginning, you need to be familiar with definition of terms like sterilization, disinfectant, antiseptic, sanitizer, germicide, microbicidal agents, microbiostatic agents, and antimicrobial agents. All these terms are maybe definition you must know to understand further about microbiological methods. Then physical methods of control like principle, construction and applications of moist heat sterilization like boiling, pasteurization, fractional sterilization, tyndallization, and autoclave. So up to here, all this which comes under the physical method of control or physical method of sterilization. Next, dry heat sterilization. Yeah, under physical methods of sterilization, all these which comes under moist heat sterilization. Okay, that is nothing but by using steam, boiling, pasteurization, fractional sterilization, tyndallization, and autoclave. So all these which comes under moist heat sterilization or maybe a steam. Next one, in contrast with steam sterilization, we have dry heat sterilization where water won't be there. Okay, like incineration and hot air oven. Next, further, you need to learn about filtration techniques. There, diatomaceous earth filter. This is diatomaceous earth filter, SIDS filter, membrane filter, and HEPA filter. Okay, this HEPA you can see in the laminar airflow instrument, which comes under the filtration techniques of sterilization. Next, radiation method, ionizing radiation, gamma rays, and non-ionizing radiations, UV rays. Okay. Next, you need to understand about chemical methods like alcohols, aldehydes, phenols, halogen, metallic salts, quaternary ammonium compound, and sterilizing gases as antimicrobial agent. So this is all about unit 1 and unit 2. Next, unit 3 is microbiological techniques, where you are studying about, learning about, understanding about culture media, like components of media, natural and synthetic media, chemically defined media, complex media, selective, differential, indicator, enriched and enriched and enrichment media. So these are the different varieties of culture media. Further, you are going to learn about pure culture methods, where you need to understand serial dilution techniques, plating methods. In plating method, you need to understand pour plate, spread plate, streak plate. Further, cultivation of microorganisms. Then maintenance and preservation, stocking of pure cultures, cultivation of anaerobic bacteria. Okay. Then stains and staining techniques. Under this, you are studying principles of staining, types of stain, simple stains, structural stains, and differential stains. Okay. Next, modes of action of antimicrobial agents, antifungal agents, amphotericin B, then grisofulvin, then antiviral agents, amantadine, then isoclopropyr, azidothymine, antibacterial agents like plasmomycin, ervacycline, omagecycline, and imipenem. Further, challenges in antimicrobial therapy, their emergence of resistance, example like MDR, XDR, you are studying. Okay, so you need to study and you should know about antifungal agents, antiviral, and antibacterial agents. Further, assessment of antimicrobial activity. In that, you are studying here antibacterial by using different techniques called DISC method or agar-well diffusion techniques. Also, microdilution method, zones of inhibition, MBC, determination of IC50, etc. Antifungal, here determination of MFC, time kill kinetics assay, sorbitol assay. Antiviral, CPE, virus yield reduction assay, TCID, neutralization assay, hematoagglutination inhibition. So these are some of the assessment of antimicrobial activity you're studying in your syllabus. So this is all about theory concept. Okay, in your in your second semester, paper 2, that is microbiological methods. Unit 1, microscopy, analytical techniques. Unit 2, sterilization techniques. Okay, definition of terms, physical methods of control, chemical methods. Then unit 3, microbiological techniques, culture media, pure culture methods, stains and staining techniques. Unit 4, antimicrobial agents and assessment of antimicrobial activity, modes of action of antimicrobial agents, assessment of antimicrobial activity. So this theory concept units you are writing in the examination for 60 marks, okay, for 2.5 hours. Further, rest of the 40 marks will be for your IA marks. This is formative assessment like assignment or field visit or project, which carries 10 marks. Okay, in that 5 marks for assignment and field report 10 marks. Okay, then test and attendance 20 marks, 10 marks for test and 10 marks for attendance. Further, participation in the class, your performance, you have behavior in the theory class, so that carries 5 marks. So total here in theory you have 40 marks for your IA. Okay, so you need to focus on this particular 40 marks to score out of out or to secure centum. I hope you understood this theory concept. Further, I'm going through the experiments you are performing in your second semester practical classes. So all these experiments in your regular practical class you are going to do. Experiment number 1, before all these 13 experiments, first you need to understand safety measures in the microbiology laboratory. So I have discussed in the previous video and even I explained, I've already sent you that video. Kindly focus on that one. First, you need to write in your record safety measures in the microbiology laboratory. Okay. Here, experiment number 1, to study the principle and applications of important instruments like biological safety cabinets, then autoclave, incubator, BOD incubator, hot air oven, light microscope, pH meter. So all these instrument principle and standard operating procedure, that is SOP, you need to understand. Okay, how to use all these instruments also you must know. Then all these instruments used in the lab, microbiology and biotechnology laboratory, and you must be familiar with all these instruments. All right. Then experiment number 2, sterilization of media using autoclave and assessment for sterility. Okay. Experiment number 3, sterilization of glasswares using hot air oven and assessment for sterility. Experiment number 4, sterilization of heat sensitive material by membrane filtration and assessment for sterility. Experiment number 5, preparation of culture media for bacteria, fungi, and their cultivation. Okay, it means you are going to prepare media for the isolation of these bacteria, fungi. Okay, then 6th one, plating techniques, spread plate, pour plate, and streak plate. 7th experiment, isolation of bacteria and fungi from soil, water, and air. 8th one, study of rhizopus, penicillium, aspergillus using temporary mounds. Then 9th one, colony characteristic study of bacteria from air exposure plate. 10th one, staining techniques. For bacteria, you are using gram staining technique. You are performing gram staining technique, negative staining, capsule staining, endospore staining, and fungal staining by using lactophenol cotton blue. Okay. Then 11th one, water analysis. You are performing MPN test to check the water, whether water is contaminated or not. 12th experiment, biochemical test, that is IMBIC. Then starch hydrolysis, catalase test, then gelatin hydrolysis. So all these tests you are understanding and studying and performing in biochemical tests. Then 13th one, bacterial cell motility, that is hanging drop technique. So there are total 13 experiments are there. You need to focus all these 13 experiments in your 2nd semester microbiological techniques. Okay. And I request you all to kindly write all these experiments in your practical observation book. Okay. In your practical observation book, left side, there will be an experiment called page will be there. No, there you need to write all these 13 experiment. As in, when we complete the experiment, kindly make one particular tick, or you can use highlighter so that the completed experiments you will be familiar. Okay. So this is all about the list of practical experiments you are performing in this semester. So in the practical examination, you are writing for 25 marks. Okay. And in practical, also you have IA marks for 25. So practical examination, you are performing for 25 and the internal assessment marks is 25. So that practical IA marks, I'm giving for the assignment 10 marks, then test 10 marks, then participation in the class or maybe attendance 5 marks. So total 25 marks. So 25 marks attendance and 25 marks practical examination. So during theory, the teacher may use lecture concept, chalk and talk, okay, then presentations by using PPT, or they may also show videos related to the microbiology units, and also assignments can also be given by the teacher, or weekly formative assessment test can also be given. So teacher can go through all these pedagogy or maybe study tools. Okay, we can use all these study tools and we can explain the concept. This is the pedagogy. Then for microbiology, you can refer the given textbook. Okay, so if you find in the library, we can refer all this. Almost all books are available in the library, central library. You can check Principles of Microbiology 2nd edition by Brown Publishers. You can check. Okay, you can take a screenshot, make a note, and you can search these books in library, or also you can refer online journals or online whatever the books available there, that also you can refer. If you want, you can purchase also the given reference books. Also here also there are some of the list of books are there. Refer these books also to be familiar with the microbiology concept. Okay, and here some of the very important books I have given here. There are total 10 books have given here. Microbiology Pelzer book is like a Bible of microbiology. If you want, even you can purchase, okay? Microbiology Pelzer. Then Microbiology Concept Applications by Paul. Even this book is also good. When compared to all the books, this Microbiology by Pelzer is really very good. Then you have here Fundamentals of Microbiology, then Microbiology by Ronald. Okay. Then Introductory Biotechnology, R.B. Singh is there. And Industrial Microbiology is also, you can refer Fundamentals of Bacteriology. Then Frontiers in Microbial Technology. Okay. And so many. And even this General Microbiology CB Power is also very important. Even if you're unable to get this Pelzar book, you can refer College Microbiology. Volume 1 and Volume 2 is the best book. You can almost all concepts you are going to get in College Microbiology by Sundarajan. Okay. Or you can also purchase this Microbiology Pelza, which is quite costly. If possible, you purchase. Otherwise, you will also get online. You can refer those content for your microbiology assignments and seminars and to refer. Okay. Same thing I've mentioned here. Kindly visit your department library or central library of the college and be familiar with microbiology concepts. If any doubts, please reach to the department and we can also discuss with me and also other teachers. Thank you, everyone. I hope you understood today's concept of theory syllabus and practical syllabus. Any doubt, please reach to me. And I request everyone, you be the gift to the world so that everybody love your presence. Thank you so much for being with me. Thank you, one and all.